We'll move on to our bot patch teams. I will start with Curi Bio's presentation on human data for next generation medicines. Thank you.
How do I, is this connected? Okay, cool. Okay. Morning, everybody. My name is Nick Geisse. I'm from Curi Bio, based in Seattle, Washington, which has very similar weather to what you're seeing outside here. So thank you for making me feel very much at home with all the jet lag. We are a company spin out of the University of Washington, and as Pasley mentioned, we are working on the botulinum toxin potency assay. Our philosophy is really about commercialization to the point where these kinds of tools are very easily usable by folks in pharma, in the pharma industry. And what I mean by that is people like me, right? So back when I had hair and a flatter belly, I was a young, fresh-faced postdoc who was a biologist, pharmacologist by training, going to an engineering school, trying to do 3D bioengineering. And they said, here's a key to the clean room. Here's your MATLAB license, get to work. And I've had absolutely no idea how to do any of that stuff. And we have found that the folks who need these kinds of tools, who need to use these types of tools every day, don't have the kinds of training to make these kinds of complex bioengineered systems. So the way we approach things is really a holistic platform way. We call it cells, systems, and data. And you don't have to read all the words on here, but basically what we're trying to do is really provide the end user with all of the tools to make them successful. So they don't need to buy the cells or make the cells themselves, then get the bioengineering part from another vendor, and then the data analysis and the instrument from another vendor, and try to shoehorn all that in and get frustrated after 6 months and say, this bioengineering stuff is nonsense. We really are making all this stuff very easy for people to use. And the end result is human functional data that'll allow them to drive typically a business decision. And we have focused on excitable and contractile tissues. And what I mean by that is basically muscle and nerve. And here's kind of an example of what we're trying to approach, right? So at the top is a mouse EMG. Sorry if you're a little bit squeamish with the photos. I'm a 2-dog owner, so it's a bit hard to look at this sometimes. And typically to do these types of experiments, you do ex vivo muscle, and you will make the muscle contract and do all sorts of measurements and look at how much force is generated. That's how you get a functional measurement typically. You can do this a lot in the dog for a lot of the diseases that we work on, as well as in humans in the clinic. You can use this to diagnose things. Our approach is to use, hopefully this mouse works, engineered muscle tissues, which have been around for quite a while, actually. But we, as you'll see, we take a different approach and actually enable people to make these fairly easily in their laboratory. This is a 2-post system. These muscles contract. As the post displaces, we can actually read where it's, how much it's moving and how much force is produced. And we can get the kinds of data, as you see at the bottom, very similar to the mouse EMG. The mouse EMG you see at the top, the kind of taller lines are from a healthy mouse. The lower lines are from a mouse that has a DMD phenotype. Down below is an iPSC line. The green line is the iPSC line when it's healthy. The blue line is the iPSC line when we knock out dystrophin, which is the protein that's missing in muscular dystrophy. So those are the types of studies we can do, and we use this for drug dosing and drug studies like that. So when I say cell systems data, what do we do? So we package this all as a single instrumentation platform with a cell culture device as well as cells. The idea is to make it dead easy, right? So we call it making popsicles with the kids. If you've ever done that as a kid or with your kids, you take an ice cube tray, you put fruit juice, you put the saran wrap, and you put toothpicks, you freeze it, pop it out, it's a popsicle. We do the same, obviously much more sophisticated, but with cells and hydrogels, and we dip these 2 poles into a casting trench. We can pull these muscle out. Long story short, we can take typically anyone who is a graduate from an undergraduate biology program who knows how to use a pipette and already has sterile technique. Typically takes about 8 hours over 2 days to train them how to do this type of experiment with 100% casting success rate every single time. So we've really focused on reproducibility and usability. When we do these kinds of things, looking down at the microscope, you can see we get a very nice uniform tissues. Typically, the variation across tissues is only a few % with respect to contractility. Again, if your pipette skills are good enough to make sure you have the same amount of cells in there. Typically, the weak point is the person behind the pipette, and it's really not that big of a deal. At the end of the talk, I'll show you how we're eliminating that. And then from there, we actually get these functional data traces. We can actually watch these things in real time and look at the contractility, see every single beat, every single movement of either the skeletal muscle or the cardiac muscle, and then we can plot these types of things and start to measure strength. So we have been working on the system for about 5 years now. Again, we have multiple versions of this system out in the world in various pharma. I have a couple customers in here. Morning. We have been focused on disease modeling. Now we are transitioning to the neuromuscular junction. As a lot of you folks know, the neuromuscular junction is a really, really difficult structure to model in vitro. Part of it is because you need to take a really long time to actually form these things in vitro, and a lot of these cultures don't live very long. Our 3D cultures can live for months to years, so we can actually have that time to let that slow biology develop. We can make these neuromuscular junctions so we can actually start to do things like model animals. And part of the goal of this particular project is Botox. So if you've ever had Botox, I haven't, but I know I've met a lot more people this past year who've done it. It's a toxin that basically stops the conduction from the nerve to the muscle, right? And so somehow that makes you nice and smooth. Turns out more than 50% of the Botox made in the world is actually for therapeutic purposes. Like kids with Duchenne muscular dystrophy, for example, they tend to get really bad contractures, and it's actually quite painful. And one way to alleviate that problem is to inject them with Botox or botulinum toxin, and that relieves some of the pain. So when you make this stuff, it's a natural product, right? So you've got these big factories, these big lyophilizers. You make a gram of protein. You're not exactly sure how strong it is. Kind of like making a whiskey or a wine. You're not exactly sure what the proof is. So you got to, oh no, my animation didn't work. Shoot, you missed the reveal. So the way you typically do these types of things is you take a bunch of mice, you get someone to inject them, and you watch them die anywhere between 1 to 6 days, depending on how you're doing it. And wherever it stops, there's your LD50 curve, there's your IC50. Okay, that's how strong it is. And the variation can be pretty high. So if I'm a single user, if Pasley gave me her test article, and if I tested it on, like last week and I tested this week, the variation in the answer could be anywhere between 20 to 30%. If I test it and then she sends a sample to Mark and Mark tests it in his lab, the answer can vary by 50%. So these tests are really, really inaccurate. And that's one of the issues that we've been really kind of trying to address here with reproducibility. So we've kind of adapted our system here. This is really complicated. I won't go through it. It's already been published anyway. But basically with the casting trench, we have these neurospheres here at the bottom. They actually innervate into the tissues. And then we can actually control the contraction. So these, what you'll see here is these kind of little spheres are going to be decorating. Actually, got a better photo here. This isn't projecting super well, but you can see those kind of lumpy spots. Those are the neurospheres that are sitting on top of the kind of little worm-like structure. These things are a few millimeters big. You can actually see them like as you hold them up. And here we've preferentially marked them with a fluorescent protein so you can actually see how they decorate there. And in this case, what we've done is we've modified those neuronal cells so they're optogenetically sensitive. So what we can do now is we can actually shine a light, and it's really subtle here. They took this early on in the experiment. Unfortunately, it's usually much more dramatic, but once we actually start flashing, you can actually see that concomitant with the flash, you see a bit of a contraction. So that's how we preferentially just hit the nerve without having to put a big electric field that'll also get the muscle no matter what to do these kinds of things. And we end up forming a neuromuscular junction. Again, this is, several people have probably seen this already in this talk before, but we start to see proteins forming at the junction between the neurospheres and the muscle. And when we start doing the stimulation, so this is pre-treatment, so this is before we treat with any toxin. The green one is the control, so we're not going to treat that guy. The dark blue one is the guy who gets treated. After treatment, we can see we have a complete cessation of stimulated beating due to the presence of the toxin. So that's a kind of simple version of doing it. Now that we have an entire 24-well dish, in this case, we can actually start to do these on a much larger scale. We can start to treat. These are all the different muscle cells before we actually, or the muscle tissues before we do the actual experiment. We hit it with the toxin, and within 24 hours, we see highest concentrations dead. As we go to lighter concentrations of the drug, we start to see beating. And from there, we can start to get things like dose response curves and things like that. This is really a summary slide. And unfortunately, I gave you the older version of this, but the EC50s that we're getting, we now have the point where we have multiple users in multiple facilities with multiple different instruments, 4 or 5 different iPSC differentiation runs, and the variability we get is on order of 3 to 3.5% with respect to the actual measured EC50 of the drug. So we're starting to get very nice reproducibility. And we're now having some customers in Asia and some customers in Europe who are doing these types of experiments. So we can now do kind of intercontinental variability type experiments too. So we're also, the new stuff now here for the consortium people is that we have been working on making the system more flexible. So we can now bring your own biology. So we have customers who actually want to do disease modeling or have their own kind of cell lines that they want to do these types of experiments with. So we're now starting to do basically a viral delivery of the protein that causes these tissues to become light sensitive. So we can start to do, so this is a plate map here. So you can, basically you're going to be looking at these tissues here that get the highest dose of the virus. And we can start to see here that, yes, we can actually deliver that protein. We can make these light sensitive. So this was basically an iPSC-derived neuronal cell line. We can now transform it to actually make it usable in this type of system. So now we can stimulate that new cell line that we've created and make it contract with the muscles. And we see this in kind of a dose response-like fashion too. Here's a little more data on that. Just kind of showing again, we can control the level of innervation and the level of sensitivity to the blue light. This is a really short talk, so I have to wrap up now, sorry. So anyway, this is the wrong slide. Oh no, this is the wrong, shoot. This is the wrong presentation. I sent them the link while I was on the plane, so I think this got a bit jumbled up. The last thing I was going to show was, as part of our qualification plan, we're trying to eliminate the human. We're trying to eliminate the variability. So we've now gone to robotic casting on a 96 well system. So now we can actually just say, hey, here, put your tube of cells here, put your extracellular matrix here, push a couple buttons, and let it go. Typically, when I think of robotics, I always think of like those $400,000 to $1 million big systems with all the arms and stuff like that. That's not what we're talking about here. We typically now use an Integra pipetta robot, probably like $20,000 or something like that retail. And basically you just push a button and it will actually cast all the tissues for you, makes your 8-hour day a little bit shorter. So from there, we're really, as we saw on the earlier talks today, really focused on the SOPs, qualification and variability across users and across space. The eventual goal is to have the customers run it. Basically, we're not going to be running it here. We're going to be shipping out the instrument and the biology and the plates to the folks so they can actually do it at their own facility in their own factory as they make these different lots of Botox. So you don't have to spend a week killing a bunch of mice. You just spend a day with a pipette and you're good to go. So that's it. Thank you.